everyone. So I study how cancer begins, and equally important, how it often doesn't. So we've known for a very long time that cancer is at its root a genetic disease. So cells acquire these driver mutations. We can then allow them to acquire uncontrolled proliferation. And what we've come to appreciate more recently is that although these driver mutations are required, they are often not sufficient for cancer development. And what I mean by that is that usually for a cancer to start growing, we need two things. So one is a cell with a driver mutation, and second is some sort of tissue perturbation or stimulus that comes from the microenvironment. This tissue perturbation often comes in the form of inflammation or tissue injury, which then allows this mutant cell to expand and switch on this program of tumor formation. Of course, at homeostasis, these mutant cells don't really have these signals from the environment, so they are unable to do that. So if you consider your own skin right now, as we age, we accumulate millions of cells that carry mutations in genes associated with cancer. In fact, by middle age, approximately 1 out of 4 cells in your epidermis harbor mutations in cancer-associated genes. And I think and I hope that no one in this room currently has a carcinoma growing on their skin. So I'm really interested in understanding what are the dynamics between mutant and wild-type cells when inflammation hits, really mimicking the very early stages of tumor development. And for that, I use the skin as a model system. And the skin is a particularly interesting model because it's our barrier to the outside world. So we're constantly exposed to different environmental insults, such as UV radiation, wounding, pathogens, allergens, all of which cause inflammation, sometimes mutations, or both. And it's really a tissue where mutant cells and inflammatory signals routinely coexist. So what I'm doing in my research is to use genetically engineered mouse models to recreate this mosaic epidermis so that we can then challenge it with inflammatory agents and tumor-promoting agents to see how these wild-type and mutant cells behave, to try and explain why mutant cells don't always give rise to cancer, and what are the cell intrinsic and extrinsic mechanisms that are behind this process. So you can come to my poster if you'd like to know more. Poster 21, I think.
Thank you.